Hello and welcome to Smith and Sheridan on Biotech, a podcast on the science and business of biotechnology, presented by me, Cormac Sheridan.
And me, Andy Smith. Hello, Cormac.
Hello, Andy. How are you doing?
Fine, thanks.
Today, we're going to talk about a category of drugs that are many decades old. And if they were invented in the last decade, we will be talking about them as wonder drugs. They save lives, they cure disease, they don't just manage the symptoms, they actually eliminate disease, and they cost a couple of hundred, maybe a couple of $1,000, and yet they are neglected by most of the biotech and pharmaceutical industry. We are, of course, talking about
antibiotics, antimicrobials.
Antimicrobials. Very, very important category of medicine that is only barely touched on because the pharmaceutical industry and the biotech industry are sort of addicted to drugs for chronic conditions that involve lifelong dosing at extraordinarily high costs that can maybe keep disease symptoms at bay, but never cure it. It's a perfect financial model. It's a very imperfect medical model. Andy, as a sort of an analyst who's worked in industry and who also looks at the sort of financial or business side of biotech, we all know the sort of problem, but maybe set it out for us, and we can also maybe explore whether there are any solutions or not. But you've talked about it being the paradox, which we all know about. But give us your kind of take on where we're at with that at the moment.
Yeah, and it's very true what you say. I mean, if they were invented today, they would be a panacea. And it's fair to say at the time when they were first discovered, I mean, when penicillin was first discovered just after the war, in fact, during the war, but then it really commercialized just after the war, it was a panacea. You know, there were infections that ultimately were fatal to many thousands of people that were effectively cured. And after the war, again, in the 50s and 60s, again, infections that the golden age of antibiotics at that stage got to change people's perceptions on, we could perhaps have got a bit blasé that medicine can sort anything out at that time. And what's happened since is you can knock the bacteria down for a while, but it's a bit like as we saw in the pandemic. It's a numbers game, and it's a large numbers game. So, you know, in the pandemic, we saw the coronavirus, COVID-19 virus, eventually evolving because of so many millions or billions of infections to actually become less affected by the vaccines. So they have mutations. So the more infections you have, the more chances you have is a breakthrough mutation, and that results in a lower level of protection. The same has been true multiple times, not just because of vaccines, but because of a wide range of antimicrobial agents. And this is not just one vaccine we're talking about. You know, we're talking about the penicillins, the chemotherapeutic agents like the sulfonamides, nitrofurantoin, glyphosate, a wide range of antibiotics and antimicrobial agents. Yet, because there are so many infections and they're all treated, and a patient with a fulminant bacterial infection will have hundreds of millions of bacteria floating around in their circulation. And almost all of them, if it's a sensitive bacteria, will be treated by or be killed by an effective agent. But there are so many that there will be the odd few mutated organisms that then become resistant. Therefore, they're selected for resistance, and potentially they can transfer that resistance over time and go on to affect someone else. Yeah. But as you say, the antibiotic premise is, well, the problems with that antibiotic premise or that antibiotic promise, the paradox of the promise and the premise, is that given enough infections, it hasn't been known yet that there is an antimicrobial agent or bacteria won't develop resistance to it. On the other side of the coin, they have been going for so long. So the penicillin, the average wholesale acquisition cost for a course of antibiotics is not in the hundreds of dollars, it's in the tens of dollars. I actually talked to a hospital pharmacist a couple of years ago who quoted me the prices that his pharmacy paid, and it was virtually pennies. So they are, for patients who have infections that are sensitive to antibiotics, they are the golden bullet. I mean, for pennies, you can cure that, and a patient in a short space of time, and that's the important thing, a short space of time, patient can then recover and go home. The reason is, the paradox is that they're treated for a matter of days. There are some bacterial infections that need more chronic antibiotic, antimicrobial treatment, like tuberculosis, like bacterial endocarditis, but mostly within 10 to 14 days, the patient's cured and is healthy and can go home. And you can't make a business model out of something. You can't charge something enough for an antibiotic, because most infections still today are by bacteria that can be treated by an antibiotic or antimicrobial agent, treated by a generic antimicrobial agent, as we already discussed, that are cheap, pennies. Now, when you then say, okay, so the small number of patients who have resistant infections, they need to be treated by new antibiotics. New antibiotics need to be developed to treat those patients. That's fine, but it's a very small number of patients. I mean, it's the thousands, 32,000 deaths, I think, in the US every year, but that's still, of the number of infections, still a small number of patients. Also, for biotechnology companies, it's very difficult to find those patients to do a clinical trial.
Although I have spoken to the developer preclinical, or no, they're an early stage development company, and that 35,000 figure you've quoted, they would argue that it's actually a massive underestimate of the real problem, because those deaths aren't properly recorded, and that the reality of multi-drug resistant pathogens in hospitals is much, much larger than that. I don't think I'm open to correction on this actually, but I haven't heard people linking death from sepsis, which is a very common cause of death, to systemic infection with multi-drug resistant pathogens. It wouldn't surprise me in the least, though, if they were a major culprit in the area of sepsis, which is one of the leading causes of deaths in hospitals. And it's not that there's some sort of conspiracy to cover up the cause of death. It's more that there's less awareness, less testing, and that very often the cause of death is given for more generic reasons, but that very often there might be a multi-drug resistant pathogen involved, but it may not have necessarily been identified. So I think the problem is actually bigger than 35,000, in my opinion.
Yeah, there are some nuances there, Cormac. A little old lady who gets admitted to casualty emergency room, then is admitted to intensive care with, say, a bacterial pneumonia or a bacterial blood infection, then potentially they can be admitted with a resistant infection. Almost certainly these days will be resistant to penicillin. It might be resistant to macrolides, but that doesn't mean there isn't an armory of antibiotics that could be used to treat those patients. But as you move up from the first generation community administered antibiotics like the penicillins, cephalosporins, the macrolides, then you get to things like the aminoglycosides and things like vancomycin that are a bit more toxic. In fact, quite a lot more toxic than penicillin. You can take grams of penicillin, and if you're not hypersensitive, you don't have a problem with it. You might have a bit of diarrhea, but you don't have any other problems. But you treat with high doses of vancomycin, high doses of aminoglycoside, you've got nephritis, they're toxic to the kidney. And that may actually be a cause of death in an otherwise frail patient.
Yeah.
So that's why the numbers are misleading. But the other factor I forgot to mention is that anti-infective physicians, ID physicians, infectious disease physicians, are well aware of the resistance problems, not just in the hospital, but with patients they see being admitted from the community. And they will, it's called antibiotic stewardship, or they will spare. There are things in hospitals called 24-hour stop orders. So if a attending physician prescribes a sledgehammer antibiotic normally reserved for resistant infections to treat a patient that the lab finds the next morning is sensitive to erythromycin or penicillins, then they have these things called 24-hour stop orders. So that initial prescription is stopped and they're put on the much cheaper agent will quite easily treat the infection. So this antibiotic stewardship means that the end market for a new antibiotic, should one be developed, is also much smaller, because there's a significant discipline these days on prescribing antibiotics.
And I can't say I've tracked this systemically at all, but has any recently introduced antibacterial attained blockbuster status or anything near to blockbuster status?
No, and that was the problem. When I worked in a pharma company, we would have people despairing at the fact that I would be the commercial person, and they'd say, go away and work out how much this product was worth. I'd come back and say, a couple of $100 million. And they say, that's not worth it. Well, it's not worth it because if you've got restricted prescriptions in hospitals. So they'd say, okay, fine. What about a community antibiotic? Well, there is much lower resistance in communities than there is in hospitals. So generally, a large number of antibiotics that are now cheap and generic still work in the community and are happily prescribed by primary care physicians to patients with a urinary tract infection or a lung infection or something, and they can be effectively treated. And as you say, that decades of antibiotic development has, for another reason, for the fact that they've already done and are still doing really good jobs, that has stymied the development of new antimicrobial agents by biotechnology companies. And we've seen companies that have brought products to the market, and because the FDA has said that's fine, but you've done a study in resistant organisms, so don't treat a penicillin-sensitive infection with something that's resistant infection. So the label is narrowed, the clinical trial costs are higher, and the end market is much lower. And that's sort of a recipe for unviable product.
Yes. And I mean, we were discussing any number of examples of recent casualties, companies that successfully developed products all the way, or at least nearly all the way, and ran out of road, ran out of cash, were acquired, assets may have been developed further or abandoned. Are there any particular examples that you think exemplify the kind of problem that the biotech companies are facing here?
Yeah, I talked to the people at the Wellcome Trust a number of years ago who were very keen on a company called Ayogen, who developed a new antibiotic for hospital infections. And it went all the way through to the market. It got approved by the FDA, and its first year or two on the market was essentially a commercial failure because it sat there in pharmacies being reserved for those patients who had the most resistant infections, couldn't be treated by any other agent. And there just aren't enough of them to make that viable. And that company, amongst many others, went into liquidation. But
you're talking about Zemdry, right, or plasmamycin, which was an aminoglycoside.
And that's the other aspect of it. Aminoglycosides have been around for many years. So have the penicillins, the cephalosporins, the vancomycins. It's going to take a lot of research work to find a new class of antibiotics because we've been at this for so long. I remember when I was at SmithKline, we had the robots running 24 hours a day, and there are, I won't name names, but there are pharmaceutical companies that had the gene sequence of Staphylococcus aureus cloned before it was ever published. So the open reading frames were expressed. They were determined which ones were essential for viability. And you just let the robots go at them, have at them, and screen the whole of chemical space for new antibiotics. And surprisingly, even to a cynic like me, we found a few things, potentially brand new antibiotics. Yet, to use the Domestos example, they killed mice just by looking at them. So it's quite hard 80 years on, right? 90 years on from the golden age of antibiotics to find a new one. And then if you do find a new one, you've got a very small market. And that pushes us towards, this is not an endeavor for a commercial biotech company. It's more of an endeavor for some charitable organization or governments to help that. And there are piecemeal efforts and germination of efforts to get to that.
Yeah, the US military is doing a certain amount of spending here, isn't it?
I mean, yes, and I applaud that aspect. I mean, I think you think back to the First World War, and I don't know percentage, but so many servicemen died in the trenches of what was at that time unpreventable infections or untreatable infections that nowadays would be treatable. And even now, if you think about the bioterrorist aspect of anthrax, is largely sensitive still to antibiotics, as is the one for plague. But you would like more in your armory than you have at the moment, obviously, always. And that's probably the military's interest.
Yeah. Well, it's interesting to me. And when I was looking at some of the background reading for this, the old nugget of bacteriophage therapy kind of course, pops up, which was pursued in the Soviet Union and related countries. It kind of more or less paralleled the emergence of antibiotics. And because of the Cold War divide, I guess the phage therapy approaches, which I think had been maybe originated actually in the West in the early 20th century, but were pursued more vigorously by the Soviets and their allies. And the two sort of never really sort of crossed over. And yet there remains ongoing interest. And the US Army has backed a Korean company, Intron Biotechnology, to develop a therapy for urinary tract infections caused by E. coli or Escherichia coli, a gut organism. And then this was a deal which last year involving a US company, Locus Biosciences, who have a very interesting approach. And they got money not from the US military, but from BARDA, which is the Biomedical Advanced Research and Development Authority, which many people will know for its work in funding development of the COVID-19 vaccines. BARDA has been around for some time, obviously, and they have backed the late-stage clinical development and subsequent events around marketing approval and all the rest of it of a development stage therapy, LBPEC01, which is phage therapy engineered with a CRISPR-Cas3 construct, which it knocks out the targeted bacteria, it's E. coli again, in two ways, both by the usual cell bursting or lysis effect on the bacteria, because phage, for people who aren't familiar with the term, or phage, are basically viruses that infect bacterial cells. So they harness that natural activity of the phage, but they also engineer in this little kind of bomb that explodes in the bacterium's genome. And again, there's been serious money here, and they're in a phase 2-3 trial, which got underway about 12 months ago. The readout from that, I think, is going to be extremely interesting, because they're taking the whole concept of phage and marrying it with, obviously, this very precise and very powerful genome engineering tool, which will give it a level of precision and also selectivity and power that I'm not sure whether the other phage therapies had prior. Why have phage therapies always been on the margins to the extent that they've been?
Yeah, and I can't tell you how many companies I've seen going back 20 years as an investor. And normally they start off in Canada or somewhere else. They come to the fore and people think, and you can sort of see if you have a lawn of bacteria on an agar plate and there's one or two phages that are dropped on that, then they completely lyse the bacteria. It looks really impressive. But outside what's happening with CRISPR, they are, okay, they're viruses that infect bacteria, but they are seen by our immune system as well as foreign. So systemic administration of a bacteriophage will result in the same way as any other bacteria, hopefully in small numbers, will be inactivated, digested, identified as foreign by our immune system and neutralized. And that makes it very difficult to administer bacteriophage as therapies for bacterial infections systemically. Potentially, it's possible to have those for skin infections where you don't have the systemic exposure. Although, by the same token, you could use antimicrobial agents, which are not as active as typical antibiotics, but they're chemicals. You could do chemical disinfection for wound infections as well. So it's, again, and that wound infections versus systemic infections is one of the aspects that has hindered bacteriophage therapy over the years. But the biggest issue is the fact that we recognize them in our bodies as foreign, and therefore we eliminate them before they've even got a chance to find the bacteria and lyse them.
But I'm assuming, though, that, and I haven't the answer to this myself, but if you look like a Danish company like Sniper Biome, and they're one of the new breed of companies that are cleverly sort of integrating expertise in CRISPR-Cas9 genome engineering with microbiome expertise as well. And again, their lead program, which is in the clinic, Sniper 001, it's a cocktail of 4 CRISPR-armed phages that target and eliminate E. coli, particularly those resistant to fluoroquinolone antibiotics. And they're in phase 1 in healthy volunteers. But the end goal of this is to develop it for people with hematological cancers who are undergoing bone marrow transplants. And they're particularly susceptible to E. coli infections spilling in from the gut. And so, obviously, a company like this, they've raised 50 million euros in Series A funding. They obviously have workarounds here. And I'm not sure it's a question of you can just target it to a specific anatomical locale. I don't know whether you can pseudotype these phage particles or in some way that they're shielded from the immune system.
Well, the other issue is related to what we were talking about previously. So bone marrow transplants, and in fact, transplant patients in general, are very closely monitored, both in their lab work, but also in how they're pre-treated prior to the transplant, but also the coverage given to them, whether it's for bacterial or viral infections. CMV is a common one in bone marrow transplant patients, but they can be treated with antibiotics prophylactically. So that pushes this a new product, should it be efficacious and safe, to be only used in those patients that have infections or breakthrough infections that are resistant to their current antibiotic cover cocktail, and even the second generation that can be used, or second level treatment that can be used for those patients. Again, bone marrow patients is not a large population. I mean, it's a reasonably large population, but it's a small number of patients. Once you get on with those that have breakthrough infections that go beyond their antibiotic therapy and coverage, and then the second line therapies, it's a much smaller number of patients, and therefore that's, as we were saying, economically less viable.
Yeah, okay. And of course, having mentioned the microbiome, there's obviously in the last 20 years or so, I suppose, certainly in the last 10, a growing appreciation of the importance of microbiome for human health. And obviously, antibiotics can be a double-edged sword because they might cure you of a pathogenic infection, but they can also damage your overall health status by damaging your intestinal flora. And we're only beginning to obviously understand the composition of the microbiome and fully understand its myriad effects on human health and on human disease. And there are as well these fascinating efforts now around microbiome engineering, and how precise or imprecise that is still open to question. But I know even Jennifer Doudna herself, one of the key inventors of the CRISPR-Cas9 genome editing technology, she's involved in this extraordinary initiative with her fellow scientist at Berkeley, Jill Banfield, to apply some of these techniques for multiple purposes, not just for human health, but even for improving the methane emissions of ruminant cattle. If you can in some way engineer their biomes and do it in a way that's easy and sustainable, that they produce far less methane, letting people to continue to enjoy their steaks if they're so inclined. And obviously, there are multiple ways to try to modulate the microbiome for human health in multiple areas. And we've not seen a whole lot of success there yet. I think we've seen some maybe dietary supplements, some minor interventions. But do you think that that's going to be, I mean, there was a lot of hype there a couple of years ago. It's died down now somewhat. Is that something that is going to yield useful therapies? And I'm also wondering, and bringing it back to infection, the role of microbiome balance or homeostasis in controlling infection. Have we made much progress there, do you think?
We are making progress. And as you say, Cormac, it is an up and coming area. And for all those discovery microbiologists who looked at anti-infectives and the problems we've just talked about associated with them, they won't be out of a job now, because they can convert to being a microbiome scientist. And the microbiome issues relate to our human love-hate relationship with the bacteria that inhabit our body or rest on our body. And in many cases, the many billions of bacteria that we have in our GI tract and on our skin, they're fine. And we're fine with them as well. But there are some occasions where either some nutritional aspect results in an overgrowth, or some trauma results in those bacteria being put into a different site, or, as you were hinting at, the treatment for a different infection can result in an overgrowth of what would normally be considered a commensal bacteria to have a pathogenic effect. And this is where the study of the microbiome is coming into place. And we've had products already approved. So there has been one product already approved, numbers in development. And for full disclosure purposes, I cover a company called Destiny Pharma that is developing, has a product in phase 3 for this indication. It's called Clostridioides difficile infections. So, there are spores of that certain bacteria in most people's GI tract. Given the right opportunity, and they're not given the right opportunity because it's such a large, diverse microbiome, they never get to overgrow and produce a toxin. But if patient has broad-spectrum antibiotics, all the other antibiotics get knocked down, our bacteria get knocked down, and their commensal effect is reduced, leaving these spores of this Clostridioides thing to then regrow after those antibiotics have passed, and then result in toxin production, and that can result in death, certainly a lot of problems with the patients. And they're very difficult to treat, because you then have to have different antibiotics, which we talked about initially as more toxic and more expensive, to actually try and resolve that Clostridioides difficile infections in the bowel. So that's been the first, really. There are a number of companies other than Destiny Pharma that have products in development or have a product approved to actually address that. And that will really be the banner waver for microbiome intervention. But as you've also hinted, there are many others. So the company also that I cover as well, most people have Staphylococcus aureus, which we think about as a pathogen that in the wrong place at the wrong time can result in serious infections. But most people have it in their nose. Destiny Pharma have a product that's aimed at decolonizing the nose. So when a patient goes into surgery, they don't auto-infect their wounds. And that's a very well-known route of, it's not the surgeons, nothing, the surgeons' aseptic surgical technique is fine. So the nurses, it's Staphylococcus aureus of one or two organisms from their nose auto-infecting their wounds and resulting in wound infections. So decolonization is one route of selectively modifying or modulating your microbiome so you don't result in infection when you're susceptible.
And slightly dumb question, how do you decolonize rather than eliminate with something as strong as an antibiotic?
Well, their clinical trial ongoing looks at patients who are about to go into surgical procedure, and they swab their nose to see that whether they've got, about a third of people have Staphylococcus resident in their nose as a commensal. So they swab those patients who then are effectively at risk for a surgical infection. And you've got an antimicrobial cream that you put up both nostrils, massage in 3 times prior to surgery, and you're good to go, or hopefully you'll be good to go in phase 3.
So it's kind of a low-tech thing, yeah.
Yeah.
And the products you're referring to as well that are already approved around the microbiome and C. diff infections, we're talking primarily, I presume, about the fecal transplants, right?
Yes. Again, there's a battle, if you like, going on between biotech companies that want to do a microbiome approach to intervene in CDIs, Clostridioides difficile infections, and those that in other parts of the world outside the US mainly have that as standard of care. But there are, as many of the companies point out, lots of problems with fecal transplants because they've got to come from healthy volunteers. And that definition of healthy volunteers is patients who don't have a pathogen in their bowel that you can transfer that material into another person's bowel and they will not get an infection. And as we just sort of hinted, one person's commensal is another person's pathogen. So it's difficult. There's lots of screening, quite expensive and potentially litigious aspects to actually doing a fecal transplant and it not working out.
Yeah, and defining what constitutes the pharmaceutical product is going to be...
That's a very good point, Cormac, because there are professors who've made their careers in looking at the bacterial species in human bowels and going so far as saying there are so many hundreds or thousands that still haven't been cultured. They can detect their DNA and it's different to the DNA of known species, but it's the unknown unknowns, if you like, the microbiome is that what makes up our commensal bacteria and what makes up the pathogens. It's difficult.
But all these difficulties can be surmounted, of course, with reproducible data from the clinic. And if there's evidence of safety and efficacy and it's reproducible in several settings, you have yourself a promising product at least, and then you build out the evidence base. Because I do see these extraordinarily sophisticated approaches emerging, using Jill Banfield working with Jennifer Doudna, one of the key pioneers of metagenomics. And this work is only beginning. It's incredibly complex, and I don't know where it's going to lead, but I think it's going to deliver some very interesting insights and hopefully products. So we've looked at one of the world's biggest, I think, medical problems of the next several decades. We haven't really solved the conundrum, and nor could we. But I suppose the final question is, say, you've painted a fairly bleak picture in the sense that the financial incentives are not present for a profit-orientated industry to properly take on this problem. And as you were saying, there's piecemeal approaches elsewhere for funding it. Is there anything that gives you grounds for optimism, or are we still in the slough of despond when it comes to antibacterials?
I think it's fair to say that there is a tall hill to climb for a new anti-infectious agent. However, come the time, come the man, or come the sector, if you like, look how the pharmaceutical and the biotech sector responded to the pandemic. There was never been an mRNA vaccine before, and then there was, and the world has billions of lives have been saved for that. So should that need be bigger more important or raising some sort of profile, then the sector can respond to that. And I have no doubt that the sector can respond to it, because in the same way as they responded to the pandemic.
Yeah, yeah. I think that's a reasonable point. I think that the scientific capability is there, and it's just a question of harnessing it. Okay. Well, in the meantime, stay safe and from infection and other threats to human health. Until the next time, bye from me.
And bye for me.
Take care.
Bye.